In this video, we will discuss about how the T-cell based immunotherapy can be used against solid malignancy. So basically what they have find out from the experiments, this experimental results is that they are able to find out one of the critical immunosuppressant effect in the CAR T cells when they are used against ovarian cancer cells, and how this thing can be overcome. So before moving into this our current topic, let me correct some of the points that I have discussed in the previous video, that is about panel L and M. Panel L is about this OA. OA represent the oleic acids. Oleic acids are usually high in the actual tumor conditions in the ovarian near the ovarian cancer cells. So they wanted to check whether it is increase in the uh, oleic acids will cause the same effect as they have seen with overexpression of this TAG-LN2 mRNA. And what they have found out is that with increase in this oleic acid expressions, they see the similar results. That is, they increase in the CD44 and Ki67 markers. At the same time, they can see that when this oleic acid is not used, they see the reduced signals for this TAG-LN2 wild type cells that we can see here and in the flow cytometer results. And when they use TAG uh, and when they use these oleic acids, they see that increased expressions of this mitotracker deep red, or we can say that increased expression of this CAR T cells that can be seen here when these oleic acids are overexpressed. And this is the quantitative analysis of flow cytometers, where we can see that the oleic acids are absent, the signal is low, whereas when the oleic acids are present, the signal is high, comparable to the control case. This indicates that with increase in the fatty acid around the surrounding, such as oleic acids, it can still take the oleic acids and give the same results as TAG-LN2 are expressed on the cell surface. So after correcting these 2 points, let us start with this today's topic, that is how this T-cell based immunotherapy can be used against solid malignancy such as ovarian cancer cells. So first they check these CAR T cells and their effect on the tumor cells, that is how much tumor cells that they are able to kill. And they can see that in the control mRNA case, that is where TAG-LN2 is not overexpressed, they see that it can kill around 36.3% of the CAR T cells, whereas when this ER stress is introduced in the control mRNA case, this things goes down. This that cells in that cells goes down from 36.3% to 8.74%. And when they use the TAG-LN2 mRNA in the control case, it's when they overexpress TAG-LN2 mRNA in the CAR T cells, they see almost the similar amount of dead cells, that is 29.8%, slightly reduced amount of dead cells. However, when they use this ER stress, in that case what they can see that when they use the ER stress and this TAG-LN2 overexpressions, the amount of dead cells increase, indicates that ER stress is been compensated or override by this TAG-LN2 overexpressions, as ER stress will reduce TAG-LN2 overexpression, but we are externally increasing this TAG-LN2 overexpression on the cell surface, so that will increase its cytotoxicity effect and cause more tumor cell death. And these things are quantified on the right hand side, where we can see that when the TAG-LN2 is overexpressed and the ER stress is also there, we do not see too much reductions in this PI annexin V signal cells or dead cells. So once validating that this TAG-LN2 overexpressions is directly related to dead cells, they designed a new CAR T cells that has this TAGLN2 DNA. It has this TAGLN2 DNA, and this CAR T cells, they called it as SIR TAGLN2 RV, and they did the experiments in the following way that is shown in the panel C, where they inject these CAR T cells and then use the analysis at 7 days, 14 days, 28 days, 35 days. And what they have find out is that in the case where they use the modified CAR T cells, that is SIR TAGLN2, they see that increased overexpression of this TAGLN2, and at both conditions, that is at peritoneal lavage and also at opentum lavage. And these things are quantified on the right hand side, where we can see that the increase in the TAGLN2 expressions in this peritoneal and omentum case. And thereafter they check the effect. They check this cell death in those cases, and what they find out is that cell death is comparatively higher when they use this TAGLN2 T cells compared to the normal SIR T cells in both the cases, in peritoneal lavage and also in the opentum. These things are quantified on the right hand side, where we can see that it's this increase is statistically significant. It is less than 0.05, and for the opentum it is less than 0.0. It is around 0.0001. And then what they check the surface expressions and the total expressions of the FAV2 in the peritoneal lavage and opentum, and they can see the surface expressions are increased whereas total expressions remains the same, indicating that the most of this intercellular FAV2 is being transferred to the cell surface, as there are more overexpressions of the mRNA. And same things they have observed for the opentum also. There is an increase in the surface FAVP expressions, but total FAVP5 expressions remain same, and same things are quantified and show the same results on the right hand side, where we can see that surface expressions are high but total expressions are same for both peritoneal lavage and opentum lavage. Then they check the other factors that they have checked in the previous video. It is CD44 and not CD44, this IFN gamma has been increased in both the conditions, in both the positions, that is peritoneal lavage and opentum, and the quantifications also show the same results, and similar results are observed for this TNF and also for this granzyme, as these are high in the case where SIR TAGLN2 T cells are used compared to the SIR T cells. And they check the total flux in this panel J, that total flux of these tumor cells goes down, and they use this shown the results for 3 different conditions. First one is the untreated cells. So after 35 days in the untreated cells, we can see there more tumors, whereas in the SIR T cells we see the reduced tumor, and in the case of SIR TAGLN2 T cells we see that further reductions in the tumors. And these things are quantified in the panel K, where we can see that with increase in time, that is increase in days, we see the more flux are present in the untreated cells, and slightly reduced flux are present in the SIR T cells treated mice, and very reduced flux cells are present in the SIR TAGLN2 T cells treated mice. And also this amount of tumor cells are also reduced in both this opentum region in the opentum regions in the case of SIR TAGLN2 compared to the SIR T cells, that can be seen in the panel L. These things are quantified in the panel M, where we can see that in the case of SIR TAGLN2 the CD3 plus T cells infiltrations are higher, that is they are more present in the ovarian cancer cells or in the periphery of the ovarian cancer cells compared to the case of the SIR T cells. And next they check the survival rate in the aggressive ovarian cancer stage, aggressive ovarian cancer or ovarian cancer at the higher stage, what they have find out is that the CAR T cells is not able to increase the life survival expectancy of the mice compared to the untreated cells, whereas the TAG-LN2 T cells is able to increase the survival expectancy of the mice. That can be seen in the panel L. So all this indicates that all these results, and together with the previous results that we have discussed, indicates that there is immunosuppression occurs in the CAR T cells when they come in contact with the ovarian cancer cells, such that this FAV2 expressions goes down. That will cause them less cytotoxic effect. That will cause the T cells to have less cytotoxic effect against the tumor cells, and this thing can be overcome by using the CAR T cells with TAG-LN2 T cells, TAG-LN2 cDNA modified in the CAR T cells. This will help in overexpression of this TAG-LN2, and it is independent of the ER stress. And researchers has also checked this immunotherapy effect of this immune checkpoint, that is PD-L1 receptors, and they see that PD-L1 receptor does not have too much impact on the results of this TAG-LN2 T cells, TAG-LN2 CAR T cells based immunotherapy. So these things, that is CAR T cells with TAG-LN2 can improve the immunotherapy efficiency against the solid malignancy, and they have shown here for the ovarian cancer cells in the mice model. So I hope you understood how they made their point, how they claimed their point that the T cell based immunotherapy can be used against the solid malignancy. If you need to understand more about these experiments in detail, you can go through the reference that is provided in the description. That's all for this video. Thank you for your time.